Hi, my name is Emily Stone. Today I'll be presenting a poster entitled Facultatively Parasitic Mites as Vectors for Microbes and Arthropods. In this project, we hypothesized that M. musca mites feed on a species of flies called Drosophila melanogaster, and that while attached to these hosts, they can contract bacterial infections and then transmit these infections to new subsequent hosts during further feedings. So for our experiment, we would take adult female D. melanogaster virgins and inoculate them either with PBS as a control or Serratia marcensis bacteria at an OD of 0.6. We then allowed the flies to recover for 2 hours before introducing them to mites in an infection chamber. They were then allowed 1 hour before we checked for attachment. Mites that did not attach were discarded, and those that attached were placed into new vials while still attached to their flies and left for 24 hours to complete feeding. 1 day after our initial host feeding, mites were removed from the first vial and placed in a new vial with uninfected second virgin hosts for another hour. We then discarded unattached mites a second time, but attached mites were allowed to remain with the second host for 10 days and periodically examined for attachment and host survival. Lastly, 10 days after a secondary feeding, after feeding on their second host, mites and flies were crushed and plated on individual sides of an LB agar plate. These plates were then left in a 20 degrees Celsius environment for 24 hours to promote the growth of bacteria. Growth of Serratia marcensis colonies was indicated by a production of red pigment from plated flies or mites. So of the 97 flies we inoculated, 25 had mites attached to them. 24 of those had been inoculated with bacteria and 1 was a control. All infected flies turned pink and died within a day of inoculation, indicating a successful transmission of Serratia marcensis to these flies. Of the 25 mites that fed on our initial hosts, 21 reattached to new flies. 1 fly expired before the 10-day observation period could be completed. All other pairs survived the 10-day infection period and were then plated on the LB agar plates. 1 of these mites was also a control. Of the flies and mites plated after our second experimental cycle, 5 pairs produced red colonies indicating successful infection by Serratia marcensis in both the mite and fly specimens. So, like many labs, our research was interrupted by external events this semester. We'll be resuming this line of research as soon as we can, and we'd like to repeat our experiments to confirm their veracity, as well as introduce a few new kinds of bacteria into the system. We would also like to, in the future, examine the effects of infection on mite behavior. For example, ticks, which are closely related to these mites, have been demonstrated to exhibit behavioral changes when infected with Lyme disease. So we're wondering if something similar happens with mites when they get infected with bacteria from their hosts. Thank you.